My name is LeHdPA, and today I'm going to talk about biological nanomechanics in relation to ATP synthesis and deuterium depletion. Might be an interesting topic as a new way of looking at glycolysis and the TCA cycle, for example, for energy production. And the reason I combine the two topics is that it's really hard to explain why glycolysis is so complicated, and so is the tricarboxylic acid cycle. For example, in glycolysis, we cleave a glucose molecule into two pyruvate molecules, and it takes 10 enzymatic steps, which seems a little bit complicated. And the same is true with the TCA cycle. From citric acid, we cut off, or our mitochondria cut off 2 CO2 molecules, and it takes 9 enzymatic steps. Why biology and biochemistry is so complicated when the logistics seems or would dictate a more simple approach? To understand that process, we need to look at glycolysis as a set of reactions that alters exchanges and removes hydrogens from carbons. Because if you go through the steps of glycolysis, what we see is that the end product, which is pyruvic acid, the carbons that are getting oxidized in the first and the second round of the TCA cycle are the carbons that have no hydrogen atoms anymore. And one would say this is somewhat hard to understand, as hydrogen is so important in mitochondria, as hydrogens are providing the reducing equivalent for ATP synthesis, yet the substrate in the form of glucose, by the time it gets to pyruvic acid and then acetyl coenzyme A, the carbons that are getting oxidized have no hydrogens. So it seems like glycolysis is like a metabolic dryer to remove hydrogens and hydroxyl groups. And actually, enolase reaction removes a water molecule from glucose, leaving the carbons without hydrogens. Yet when we get to the TCA cycle, what we see is water is added back to TCA cycle intermediates. This is where pyruvate comes in. And the hydrogens that are carried from glycolysis on the third carbon, they are actually lost by the time this carbon will get oxidized, and it will be replaced by hydrogens coming from the mitochondrial matrix. So the understanding of this process would require a rethinking of glycolysis and the TCA cycle, and this is what I'm going to be covering in my talk. Let's watch this movie, which is mitochondrial function. And as you can see, the energy production of ATP in the form of ATP during ATP synthesis occurs by using these proteins in the mitochondrial inner matrix, which are complexes as well as complex 5 ATP synthase. And this is the outer mitochondrial membrane, and this is the inner mitochondrial membrane. This is where these enzymes are sitting. And as you can see, there is a proton gradient as a result of complex function. Proton gradient meaning that there's hydrogen and protons pumped out from the matrix from TCA cycle substrates with the help of reduced equivalents. And when the protons are coming back one by one, you can see here there's a short tube from the inner membrane into halfway down to the inner mitochondrial membrane, and they get attached to a rotor. It's actually an electric motor, and they spin around as they are pushed in the mitochondrial matrix through another tunnel, but they have to go around and they have to turn this protein around, and in the meantime there's ATP synthesized by electromechanic forces. Now there are a lot more hydrogens and protons, better to say, in the outer membrane, and as this pumping function of the cytochromes fill this space with extra protons, which is about 250 per 1 proton, this gradient drives this electromechanic motor at very high speed. We know the speed is about 9,000 rotations per minute. We sped this up, this animation, which is from Visa Animations, and it is available from the web. Yet the important part is that this electromechanical force in the proton gradient direction from high proton concentration to low proton concentrations, by turning this protein, by attaching to a asparagine residue, there's actually continuous rotation at 5,000, 9,000 rotations per minute. Now, this is faster than a car engine. If you look at a Corvette, you can actually figure that the engine of this sports car is only turning about 6, max 7,000 rotations per minute, and ATP synthase in our mitochondria turns 9,000 rotations per minute, which is very high velocity. Actually, it's higher with a sports car. So you can imagine what type of engineering or what type of mechanics apply to this mitochondrial nanomotor to be able to perform at such a high rotations per minute. And this is why we call it nanomechanics, because these proteins are very small. They are actually in the range of nanometers or micrometers in size. And that's why we actually just look at them as nanomotors or nano-engines. But nevertheless, the process takes place in the mitochondria, and this is the inner membrane. And protons or hydrogen ions are actually carried through this short tube to the rotating part, which is called C10. And protons, one by one, get attached to the rotating protein asparagine residue, which is called Asp61. And in the static or the stator protein part, which is in the membrane, it's actually anchored in the membrane and it's not moving, has a positively charged arginine 210 residue. And when this binding site becomes positively charged by a proton, it will push the rotor to the leftward direction. And at the ATP synthesis occurs with this continuous turning of proton loading positions, these asparagine 61 residues of the membrane rotor at about 9,000 revolutions per minute, which is a very high speed and a very high revolution for a nanomotor. Now, anybody can calculate that actually by 9,000 revolutions per minute, the ATP synthase C10 subunit, and it's called C10 because there are 10 binding sites on this rotating subunit, transfers approximately 90,000 protons per minute. And that is actually 1,500 protons per second, which have to be taken up and released by this nanomotor, this nano-engine in the mitochondrial matrix. Now, hydrogen has a stable isotope called deuterium, which has a proton and a neutron in the nuclei. And it also appears in nature. And in about 1 million hydrogen molecules, among 1 million hydrogen molecules, there are about 155 deuteriums, and those are actually everywhere in glucose and any product that are produced by biosynthesis using water and hydrogen from water through photosynthesis. So these deuterium atoms can actually get incorporated into substrates just like glucose, just like hydrogen, not very big numbers, yet they have a very unique characteristic and strong isotope effects. And that is simply if a deuterium is encountered in the nanomotor, it breaks it. So actually a single deuterium can disrupt the function of the nanomotor, and this talk is about how biology deals with this process. Deuterium has a mass of 2.1 unified atomic mass units with a charge radius of 2.1 for femtometer. It's 10 to the minus 15 meter. The proton's charge radius is only 0.88 femtometer, and the mass is only 1 daltons. So you can imagine that this 1 dalton proton, when it's replaced by a deuterium in the nanomotor, it actually causes big harm to it. It's usually I describe this to my students as like somebody's playing pool and somebody would change the 8 ball to a twice as heavy 8 ball. Probably they stop playing because the rest of the balls just kind of bounce off this heavy 8 ball, and this is how deuterium behaves in biology. Actually, it's worse than the twice as heavy weight 8 ball, because actually deuterium has very severe kinetic isotope effects, which I'm going to talk about a little bit later. We retrieved this information from the U.S. government website just recently. So deuterium has 2 daltons and hydrogen has 1 dalton. It seems that this proton gradient driving the nanomotor and the very delicate protein structures are not able to withstand deuterium. And we know this because deuterium and deuterium presence in the plasma is very high. It's the maximum and the upper limit is 14 millimole per liter, which is actually the highest inorganic element in our plasma compared to calcium, magnesium, potassium, and glucose. So actually we have most of deuterium as a unique isotope in our system. And when it comes to mitochondria and deuterium don't go along too well. Actually, the deuterium-enriched water or deuterium can break the nanomotor. And to explain that, let me go to this scheme. This is the asparagine 61 rotating residue, amino acid residue, and as I told you, the protons attach to it, and then they will push around this arginine. They push the rotor to one direction, and deuterium being twice as large and twice as heavy as compared to hydrogen, they just don't go very easy through this nanotube, and once they attach to the asparagine 61 residue, because of their low acid dissociation constant, they don't get off of asparagine 61. There's the asparagine acid dissociation constant of deuterium is about 1/3 of that of hydrogen. So if deuterium occupies the asparagine 61 binding site, this rotor will have a stutter and it will break. And there are papers that just show that. So when it comes to natural deuterium abundance, for example, in glucose, every 6,600 protons or hydrogens in a glucose molecule will have 1 deuterium. And looking at the 1,500 protons per second transfer of protons, if the cells are oxidizing glucose, and there is 1,500 protons per second transferred in the mitochondrial nanomotor using this high velocity of proton transfers. If there is deuterium, natural abundance deuterium in glucose, this nanomotor may break in about every 4.4 seconds, because this is how much time it takes to actually bump into a deuterium as we are counting protons using natural enrichment. It's not a common isotope. It's a very rare isotope. It's only 0.01% of all hydrogens, but because of the high deuterium proton transfer velocity, they actually get to the nanomotor at about 4.4 seconds. And that's not good, because breaking the nanomotor at every few seconds will require a huge resource from the cell to re-synthesize all the proteins and use all the reducing power and equivalents to produce new proteins and new membranes is just really not energetically, it's just not feasible for the cells. So we believe that actually deuterium depletion is a very important, critical, natural process, biochemical process to actually save the nanomotor, and it starts with glycolysis, and this is why we have 10 reactions, because glucose has 12 hydrogens, and to strip off all hydrogens from glucose by the time it gets to the nanomotor and replace them with a matrix or metabolic water hydrogen, that's how many reactions it takes. And failing the body's natural deuterium depleting processes, we believe, is how aging and how cancer and some metabolic diseases and neurodegenerative diseases occur. And we have evidence of these. There's literature out there which explains these processes. But when it comes to mitochondria, it's important to understand that matrix water, which is formed from protons that are released into the matrix and in the presence of oxygen, this is a very critical deuterium depleted water source for the citric acid cycle to keep the TCA cycle and ATP synthesis running. It's like an oil filter in your car. It's like a precise, clean gasoline and oil supply for your car, so the nanomechanics assemblies and parts can actually work and run easily in your cells. Once we load these systems with deuterium, the nanomotor will slow down or break, and certain diseases develop. Metabolic disease epidemics, we believe, in obesity are the result of excessive deuterium extraction from sugars by carbohydrate oxidation, and because of that, matrix water and cytoplasmic water is not deuterium depleted as much as it should be. Nutritional ketosis and has had benefits by deuterium depletion would be another important result of understanding these processes as far as glycolysis is concerned. Now, how hydrogens are exchanged or removed from glucose step by step. Hexokinase would activate glucose, means that it is trapped in the cells, and isomerase exchanges from cytoplasmic water the hydrogens between carbon 2 and carbon 1. The isomerization process is not just simple hydrogen repositioning. It's actually hydrogen would form water or will be replaced from water from cytoplasmic water onto the first carbons, and then the protons are dissociating from the second carbon. This is a very important process called LeHdPA, ketose transformation. I talked about this in previous talks. I'm not going to go into details, but I just wanted to point out deuterium depletion starts immediately as our glucose is getting oxidized or glycolysis is starting in the system. Then the aldolase reaction would cleave the hydroxyacetone phosphate and glyceraldehyde 3-phosphate, and the rest of the reactions in triose phosphate isomerase, for example, performs the same water hydrogen exchanges between cytoplasmic water and glucose. So now we are replacing 4 hydrogens, or getting rid of 4 deuteriums. If they happen to be deuteriums, we are replacing them with hydrogens through phosphoglucose isomerase and triose phosphate isomerase. There are additional hydrogen losses through glyceraldehyde oxidation, and then there is a water molecule by analase taken out of glucose. So when we actually get to pyruvate, it's completely dry. And the first 2 carbons will be oxidized, and obviously they are not only dry, but we can say for sure that the carbons that are getting oxidized, will not carry any deuterium. because when they are rehydrated, or when hydrogen is added to these carbons before oxidation, those can only come from matrix water, which is deuterium depleted, as we're gonna see the various deuterium discrimination processes through glycolysis and also through the TCA cycle. But anyways, this new look of glycolysis explains why there are 10 enzymatic steps in glycolysis. It's not to generate ATP, it's not to generate lactate, it's not to generate 3-carbon metabolites. It is to generate a dehydrated, in that sense, carbons without hydrogen, which get oxidized in the TCA cycle. By the time the 3rd carbon is oxidized, these hydrogens are lost, but these hydrogens may be already deuterium depleted because of the triose phosphate isomerase reaction. So you can see that there is a lot of exchange and there is a lot of potential deuterium depletion in glycolysis, and this is why these 10 reactions are linked and channeling to one another, simply to make sure there's no hydrogen and deuterium from the environment that is carried into mitochondria. So it seems that glycolysis is a metabolic dryer, and seems that the ripping and extracellular hydrogens getting off of glucose is a very important function of glycolysis, and this is why it seems so complicated, but hopefully now it's more simple to understand. And the TCA cycle has the same, yet the opposite role, and it acts like a washer, and it hydrates, and it adds metabolic water to TCA cycle intermediates and metabolites, and I'm going to show you how. Citrate synthase uses a water molecule from the matrix, which is deuterium depleted. Then water is exchanged. Aconitase is adding another water molecule from the matrix. It's called aconic acid hydratase. And fumarate hydratase, which is fumarase, which actually yielded and resulted in the Nobel Prize in 1937 to Albert Szent-Györgyi, hydrates fumaric acid to form malic acid. And malic acid, when it's getting oxidized, it's a very important process. We'll talk about it later. When it gets oxidized, it will actually release those hydrogens and protons to NAD, and citrate is ready to accept water and exchange water, which is matrix water, to deuterium depleted matrix water to make the ATP synthase function and turn easily. So the 3 reactions which are specifically yielding and adding water to TC cycle metabolites are citrate synthase, aconitase, and fumarase, and the dry pyruvic acid, this is how it gets rehydrated in the TC cycle, as I described before, acting like a wash machine or washer after the dryer, which is glycolysis. Again, this is a very important logistics, because it makes more understandable why the reactions of the TC cycle take enzymatic steps, 9 of them, to actually operate the ATP synthase in a safe manner with deuterium in a deuterium depleted environment. So outside substrates from the environment, like glucose, cannot really drag deuterium into the mitochondria because of these safety measures in glycolysis as well as the TC cycle. So how about the ATP synthase? It seems that ATP synthase sits 2 of them together, and there seems to be a deuterium depleting or deuterium filtering protein just right at the entry of protons into the turning nanomotor, and these oligomycin sensitive proteins seem to be able to store and collect some deuterium, and we believe this is the primary function of these proteins associated with the proton entry. into the nanomotor. Now, another interesting element of the TCA cycle and ATP synthase is that how the two reactions are actually coupled, meaning that the TCA cycle turnover is providing sufficient and enough protons to run the ATP synthase, but it never exceeds the capacity of ATP synthase, or if it does, then there's an uncoupling event which is always associated with some drug action or some health, or some compromise, some defects in metabolism. Well, interestingly, these two residues, the asparagine 61, which has 1/3 of the acid dissociation coefficient with deuterium, 1/3, and arginine 210, which is the positive stator, helping the protons to push the nanomotor, are also part of malic acid dehydrogenase or malate dehydrogenase to actually turn the cycle over and yield new oxaloacetate, which can participate in the citrate synthase process. So the regeneration of oxaloacetate, which is more like the carrier of carbons in the TCA cycle, to regenerate it from malic acid, it actually takes the same residues. Asparagine 168 is responsible in pyruvate dehydrogenase for proton transfer, and the stator, or the proton fixating amino acids, are arginines, again, just like in the nanomotor. And this will make sure that the deuterium hydrogen ratio kinetically regulates the same way both malic acid dehydrogenase, which regulates the turnover of the cycle, and ATP synthase, which produces ATP using these protons that are actually removed from substrates and carried into the to the membrane cytochromes, and then getting pumped back through ATP synthase. So the same pair of amino acid residues control hydrogen proton dissociation kinetics in both enzymes, ATP synthase and malic acid dehydrogenase, which is a very key process to regulate the TCA cycle and ATP synthase in the same manner, based on the hydrogen deuterium concentration or enrichment or ratio in mitochondria. So to answer the questions that we raised in the beginning of my talk, in fact, it takes 10 enzymatic steps to exchange and remove 12 hydrogens from glucose before entering the TCA cycle as pyruvate. And this is a safety measure to make sure there is no deuterium from the substrate that gets into mitochondria, which would compromise ATP synthase function. And cleaving the 6-carbon glucose into the 2 3-carbon products, pyruvate, during glycolysis is a very important, but not the prime, and that's not the only function of glycolysis, but in the process, deuterium depletion and exchanges are very critical to maintain ATP synthase turnovers. And the same is true with the TCA cycle. It takes 9 enzymatic steps to replace 3 hydrogens with water and cut 2 CO2 molecules off in the TCA cycle, and ATP synthase needs to operate in a deuterium-depleted interfacial metabolic water environment. And this is why the matrix water gets recycled through hydrating TCA cycle intermediates, cutting off the proton, oxidizing the carbon as CO2, and then those protons return in the form of water with the help of oxygen as what we breathe in. This is the biological oxidation process, and start the deuterium depleted water recycling in and within the mitochondrial matrix and the intermembrane space, again, without allowing glucose to bring in any hydrogens. It's the same with fatty acids. Fatty acids are already depleted of deuterium, either through photosynthesis and from fat, which are low in deuterium, or if those fat molecules are in our system synthesized from glucose, they have to be synthesized from citric acid through the citric acid shuttle, which is also a TC cycle bound process. So our system, our liver, and our organs make sure that the new fatty acid synthesized from glucose is also as much as possible deuterium depleted, because pyruvic acid has to be oxidized for citrate formation, and that's key to get rid of all the hydrogens that are coming from the environment for our internally de novo synthesized fat pool. So it seems that our biochemistry and our biochemical reactions are designed to deplete deuterium. As many steps it takes, that's how many steps they will go through to make sure that our ATP synthase can work in a deuterium depleted environment. It's metabolically complicated, yet from the logistics point of view, it's very simple, because we just have to get rid of hydrogens and have to go through as many reactions as there are in glycolysis and the TC cycle. So it's a very sophisticated system. It allows the co-regulation of the TC cycle to malic acid, dehydrogenase, and ATP synthase the same way, using the same amino acid residues, asparagine and arginine, to actually respond to the same kinetic isotope effects of hydrogen versus deuterium ratio in matrix water. But we believe that this is a very critical understanding of the role of glycolysis and the TC cycle in energy production without deuterium, which breaks the ATP synthase nanomotor. So I'd like to thank Ron Penna and Dr. Q from Epigenix, Dr. Shomyahi from HID LLC, who actually first proposed that hydrogen and deuterium ratios are very critical in cell growth and cell transformation. Augie and the Ron Hertzberg Foundation funded some of the research. Dr. Lee, my colleague and my mentor at UCLA, and Esther is responsible, and she was designing the visual arts. And this is the enzyme fumarate hydratase. This is Dr. Albert Sendiody's Nobel Prize, one of them, one of the reasons why he got the Nobel Prize. And this enzyme adds water, deuterium depleted matrix water, that is generated from glucose after all the glucose hydrogens are stripped off through glycolysis, which takes about 10 reactions in glycolysis and 9 additional reactions in the TC cycle. Thank you very much for your attention.